In this lesson, we are looking at passive and active immunity. We only have one dot point, and it's an analyze the differences and similarities. All right, let's talk about types of immunity. Immunity can be classified as produced naturally or artificially, and it's not a judgment of quality, okay? Regardless of whether it's natural or artificial, it can be passive or active, just like our learning. All right, so we have a focus on antibodies here. We're talking passive and active antibodies being produced to a particular pathogen or antigen, right? And if we're talking passive, we have two kinds, and if we're talking about active, we have two kinds. So that's how we're going to classify it based on what we need to be able to do as a skill. All right, talking about passive immunity first. So if we're passive, we're accepting, we're allowing of whatever's happening to us, and we're not responding actively. And this, again, is about antibodies. So if the antibodies are acquired from somewhere else, it means our body is not actually making them themselves, okay? Antibodies can be made in different organisms, transferred to us, made in a lab, whatever, okay? Sadly, though, these don't generally last as long as those ones that we make ourselves. They degrade in the immune system, and they can't actually be replaced by the immune system. Let's talk natural first. This is all about mother to baby in some way, shape or form. So mother to fetus via the placenta if the baby is not yet been born, and mother to baby via breast milk if it has and it is breastfeeding. So we're talking always about this antibody passing. And until an infant's immune system is able to actively make antibodies, these are vital. If you follow antibody levels before and after birth, so we're talking, this is all before and after birth, you can see the passive transfer is really, really high before birth, so via that placenta, but it can take anywhere between months and years for an infant to make a comprehensive variety of antibodies on their own. All right, artificial passive immunity. Now, this is when the body, the body is still receiving antibodies, but they're made in another source, so therefore they're purposely made to give to us, right? So the antibodies can be made in a lab or in another organism. They are transferred into the body, makes sense, okay? And an example of this is snake venom. Now, I don't know much about snake venom, but I know that this is an example of artificial passive immunity, so anti-serum. Another really good example of it happening right now are COVID-19 patients whose body has, in fact, been trying to fight the disease, do make antibodies against the pathogen, do make antibodies against the antigens they see. So therefore, we would like recovered patients to be able to donate their plasma so we can collect all these antibodies and give them to those patients who are still suffering through the disease. All right, active immunity. Now, essentially, this is obviously the opposite of passive. The body does have the ability to make its own antibodies, and this can be done through training, through exposure to antigens. Our memory B and T cells contribute to this response, as you'd expect. All right, natural active immunity is pretty much everything that you know about the adaptive immune response, in particular the humoral immune response, so our B cells making those antibodies. So the antigens or the pathogens through the first line, they get through that first and second line of defense and they trigger the B cells to produce antibodies. We know that this is a trainable response, okay? The more your body becomes exposed, the more types of antibodies it's able to make. Artificial active immunity, however, it's purposefully exposing the immune system to these antibodies to do that training, okay? And vaccines do this. So vaccination, also known as immunization, and I might use those terms interchangeably. So vaccines can contain an altered or a weakened or a killed pathogen like bacteria or a virus, and it can be inactivated or weakened, can be inactivated toxins or proteins or things like that that the pathogen would produce. And these are highly specialized to the antigen that the body will actually encounter if it was exposed sort of in the wild or in a natural exposure situation. If we have a look at our different types of exposure, that first time there's a little bit of a lag before your body figures out what it needs to do and then increases. But then that second exposure, it's increasing quite considerably. So if we have this first exposure through a vaccination in a safe, controlled manner, then by the time we actually get exposed to it in the wild, then hopefully our body can mount a larger response. Okay. Immunization programs are created to ensure that individuals have comprehensive responses to a variety of antigens. Sometimes multiple vaccinations may be required in order to train the immune system, all right? It's not going to happen in the first particular one, all right? And also those memory B and T cells can start to die off after a number of years, so obviously booster shots need to happen. Now, we in Australia have a national immunisation program. Each state might have a slightly different version, and this is New Zealand's. And you know, babies don't start getting these vaccinations until about 6 weeks, mainly because the maternal antibodies can still be circulating. So if there is exposure to an antigen, those maternal antibodies are going to mop it up and eradicate it before the baby's B cells can actually respond. So probably not useful. Now, immunisation programs are ridiculously effective, okay? And some vaccines are effective with just that single dose, but others are provided in multiple doses over subsequent years. So you might have a booster after 10 years, stuff like tetanus needs that. So pathogens which evolve really quickly, like the flu, they kind of lead us to make new vaccines each year, just to keep up with the change in how they present their antigens. So flu vaccinations that we get each year are usually a mix of antigens, flu strains that they've seen a lot over the last 12 months, or those that they predict are going to increase in prevalence. It doesn't always mean that they're correct. And some years we have really bad flu years, 17 or 18 particularly bad in Queensland. If we, though, look at, say, polio, as soon as that vaccine is introduced and we have quite large coverage of people taking that vaccination, polio is eradicated. Hooray. All right, let's talk about herd immunity, because herd immunity involves this sufficient number of people in the community being immunised against this particular pathogen, okay? And it is indirect protection of susceptible members of the community and the community as a whole. It allows people who are unable to become vaccinated, infants, older people, immunocompromised people, pregnant people, you've probably heard this before, to be protected, knowing that people who can be exposed to the pathogen will be able to respond quickly and appropriately and clear it without spreading it. So it is inhibiting natural transmission. When booster vaccines aren't taken, though, this can disappear, okay? So if the herd immunity can be brought to us through vaccinations, if we remove the vaccination, we know that the herd immunity disappears. For things like whooping cough, sometimes our herd immunity disappears because people are not getting their boosters. We understand through long-term immune data that we can see that we spoke about it before, the first exposure to the antigen is a smaller one. When we get that second one, we have a more targeted response. So particularly if we're giving those vaccinations, we are going to see a longer-term response. Lots of different similarities and differences when we're talking about passive and active immunity. These are just a few, but there are far, far more. So go and do some reading, okay? Remember, our only dot point is about analysing differences and similarities.